Good morning, everyone. How are you guys doing? I'd like to welcome you all to the third day of the FDA MHRA Health Canada Joint Symposium. It is my great pleasure to kick off the Bioequivalence session today, and I highlight some of the work that we have been doing in the areas of bioavailability and bioequivalence. Today's presentations are a continuation of the discussions on important GCP topics that were discussed in the past 2 days of the symposium. However, this morning's focus will be on a specific GCP area, which is for bioequivalence studies. And that is slightly different than the conventional efficacy trial that we've heard for the past days. As you can see, this is a joint symposium with the MHRA and Health Canada and FDA. And as such, what we aim to achieve from this symposium is to share with you the collective knowledge and experiences from these agencies on inspections and regulatory oversight on bioequivalence studies, and to convey harmonized message with the hope that it helps you guys to do a facilitated drug development, global drug development. The international collaboration is one of the important strategic goals of our office, the Office of Study Integrity and Surveillance, as well as for the FDA overall. The OSIS Bioequivalence International Collaboration, specifically with the MHRA, has started more than 8 years ago. And as you can see from this symposium, the collaboration now has expanded to include Health Canada, which we started off years ago. This international collaboration has been very successful because we all work together with a common goal of making sure that the regulatory data submitted to our respective agencies are robust and reliable, and the safety of subject and participant who goes into the clinical trials are well protected. As part of the collaboration, we conduct joint or observational inspections, exchange information about the inspection details and outcomes, clarify expectations to industry on study conduct when it's necessary, and share any tools that might help the regulatory oversight. Again, by working together, we hope these efforts convey a harmonized message to industry on expectations on study conduct of bioequivalence studies across the agencies, and help us streamline the planning and execution of your own studies. As for the topics of the presentation this morning during the bioequivalence session, we have 3 very exciting sessions planned. In the first session, we will present an overview on our experience on regulatory remote evaluations. When COVID-19 hit the world at the beginning of 2020, and essentially every aspect of our lives was impacted by that, our office in FDA led an innovation and did a really groundbreaking work, which was to develop a remote evaluation approach. And it was these efforts that allowed us to continue our work and to accomplish FDA's mission of protecting public health throughout the pandemic. Among other things, we crafted a communication plan with the industry, made sure that the remote approach that we are developing is as flexible as possible given the very fluid at the time, and made the very best use of all the technologies and communication platform available, which, by the way, weren't that much at the time, as you know. Yet, we tried to make sure that we maximize the efficiency and the evaluation capability so that we can make adequate review of the study conduct and study information and make robust conclusions about the reliability of study data. Initially, we called this remote tool as a remote regulatory review, in short, RRR. Then in April 2021, FDA issued a guidance for industry entitled Remote Interactive Evaluation, RIE, for drug manufacturing and bioresearch monitoring facility during COVID-19 public health emergency. And that was the first public announcement that explained FDA use of a remote tool. Since then, the agency recognized the need for formalizing a procedure for remote evaluation across a variety of compliance programs. In July 2022, FDA issued a draft guidance titled Conducting Remote Regulatory Assessment Question and Answers. And in there, it also explained that the terminology, the Remote Regulatory Assessment, RRA, that you heard yesterday, is a category of activities that FDA uses for remote evaluation. And it encompasses the remote regulatory review and the remote interactive evaluation as types of RRAs. And as some of you may know, just a few weeks ago, FDA issued an updated draft guidance, which is now open for public comment for the 60-day comment period. In parallel to FDA's efforts, our colleagues in MHRA also developed a remote inspection paradigm. And through the collaboration that I mentioned earlier among MHRA, Health Canada, and FDA, we exchanged the tools and approaches, advantages and challenges, and notes on similarities and differences in how we conduct remote evaluations. It has been extremely valuable to learn from each other, and now I'm very excited to see how the two presentations this morning, one each from MHRA and the FDA, in the first session will inform you about the two agencies' remote approaches and provide you with the important take-home messages so that it helps you to get prepared for your future engagement and inspections by these agencies. So the next two sessions that follow, the RRA presentations, will touch on the actual bioanalytical and clinical aspects of bioequivalence studies and the perspectives from the regulatory inspections. Based on the current business models and industry trend, we see that it is generally the case that the sites that conduct the clinical phase of bioequivalence studies and those that test and analyze the biological samples from study participants are separate entities. And depending on the business relationship between those two sites, there could be more or less of communication between those sites about study design, sample collections, sample transfer logistics, and etc. And in some cases, there could be additional sites in addition to the bioanalytical and clinical sites that play various roles and functions during the study conduct. And as more entities get involved, it could easily get complicated when one tries to reconstruct all the study activities and to verify the integrity of study samples. And if there happened to be an information gap, there could be a delay in application review or even approvals. So given the pivotal nature of the bioequivalence study in generic drug approvals, you can appreciate how important it is for us to verify if subjects receive the right sequence of drug treatment and the drug concentration data submitted to agency are reliable. So for these reasons, FDA, MHRA, and Health Canada conducts inspections at both clinical facilities and bioanalytical laboratories. As for the clinical phase, we expect the bioequivalence studies are conducted per GCP principles, and documentation is complete and contemporaneous. Studies should be conducted per protocol, and all deviations should be reported. Importantly, the rights and welfare of study subject or study participant should be appropriately protected. For the bioanalytical phase, we want to ensure that the method that was used to analyze the study samples is fully validated and works robustly during sample analysis. The tools and the processes that measure drug concentration should be clearly established and properly controlled. Throughout all the phase of the study, from the subject screening and enrollment to sample analysis to data reporting, we expect that a robust quality management system be in place. So I know many of you working in the pharmaceutical field put the patient in the center of everything you do and try to do the right thing. And we and the public really appreciate that. In some cases, however, we also have seen some concerns with data from study sites. I feel that we as regulators need to let the industry know of our expectations on study conduct and best practices and pitfalls we have seen. And that is exactly why we are holding a conference like this today. We perform a number of bioequivalence inspections each year, and the presentations from the 4 speakers in the 2 sessions this morning will highlight some of the common or unique observations from our inspections that are worth noting, so that you can pay attention to those aspects when you're conducting your own study. So we have a morning that is full of important and informative presentation, as well as opportunities for you to ask questions and discuss with the esteemed panelists. I would like to express my sincere appreciation to all the speakers and moderators who worked so hard to make today's event informative and impactful. I hope that you are as excited as I am, and I look forward to having engaging interactions and lots of great discussions. Thank you so much. So I'd like to hand off the podium to the moderator of the first session, Dr. Seongeun Kasim.
Jeff warned me about that. Good morning, everybody. Good afternoon and good evening. Really glad for everyone who could meet us here in the room and everybody online virtually. My name is Seongeun Kasim. Thank you to Julia for the keynote opening and welcoming me. I'm the director of the Office of Study, Integrity, and Surveillance at the Center for Drugs and FDA, and I'll be your moderator for our first 2 sessions this morning. Today we're in for a real treat. We have 2 presentations in our first session, as Julia alluded to, focused on the use of and the development of the remotes in bioequivalence oversight, both by US-FDA and the UK MHRA. Our first speaker is Dr. May Oh, who plays a critical role in OSIS. She's the lead pharmacologist of our clinically focused BA/BE team in OSIS's Division of Generic Drug Study Integrity. After May provides an update on how OSIS developed and has been using RRAs, our second speaker is Michael McGuinness, who will provide a perspective on remotes developed and used by the UK. Michael is the head of GLP and laboratories within MHRA's Compliance Team 1 and the head of UK's Good Laboratory Practice Monitoring Authority. I'm really looking forward to their presentations and our Q&A panels after session 2. Just to remind everybody, we've switched the order of the agenda today. We're going to have our break before our first question session, so it'll be announced from the podium at the time, but just to let you know that this morning we'll take a break after session 2 and then come back and have our session with the 3 speakers that we've had from sessions 1 and 2. All right, well now please join me in welcoming Dr. Oh.
Thank you, Sean. Thank you. My name is May Oh, and I really want to thank this opportunity to give a presentation today. It works. Okay, here we go. So my presentation is Remote Regulatory Assessment, a valuable tool for OSIS to support drug application review in FDA. So this is my disclaimer. And for the overview, first, I'm going to briefly introduce who we are and what we are doing. And then OSIS oversee in vivo BA/BE studies, what we're doing for the remote regulatory assessments, and what we are looking forward for the future. So we are Office of Study Integrity and Surveillance, OSIS. So we are in OTS in Cedar. There are 2 divisions in our office, Division of New Drug Study Integrity and Division of Generic Drug Study Integrity. Other than the two divisions, we also have immediate office, project manager team, as well as CREST team. So all the team work together to support OSIS fundamental inspectional activities. Because of that, we do have several stakeholders in CEDAR, which we call review divisions. So we support Office of Clinical Pharmacology and Office of Generic Drug. We also have lots of collaboration of Office of Compliance, Office of New Drug, etc. Other than that, we do have lots of collaboration with Office of Regulatory Affairs, ORAs. So we do lots of inspections, ORAs together. So here is our office vision and mission. So for the vision, OSIS improves the public health by protecting study subjects and promoting properly conducted studies. For the mission, OSIS promote the public health by ensuring the welfare of study subjects and by verifying the quality, study integrity, and regulatory compliance of the BAB studies, GLP, and animal rule studies. So for what we are doing, simply say, we do select, evaluate, review, and support. For the select, based on review divisions consult as well as our CREST team site assessment, we do select the sites that need to be conducted inspections or RAs. After that, OSIS reviewers will perform the inspection or RAs, which is to ensure the integrity of the study data generated by those sites. So after inspection or RAs, OSIS reviewers will, based on all the evidence we collected, we generated EIRs or RA reports, as well as the OSIS review. So for all the activities, we support CEDAR review process and by give the data reliable recommendations, as well as the compliance evaluation. For the typical in vivo BAB studies that we conduct in either inspections or RAs, usually there are two components. The first is the clinical component. For the clinical inspections or RAs, they are mainly conducted by ORA staff. Sometimes OSIS reviewers also will join them for inspections or RAs. For the clinical component evaluation, we evaluate the adequacy of the site, the facilities, the personnel, procedure, subject screening, enrollment, investigational drug administration, subject safety monitoring, protocol adherence, biological sample collection, and so on. The second component of the in vivo BAB studies for inspection RA is analytical component. So for this component, usually it's OSIS reviewers conduct the inspections or RAs. And sometimes ORA staff will join us. For this component, OSIS reviewer will also evaluate the adequacy of the site, the equipment, personnel, biological sample processing, method validation, study sample analysis, documentation, data security, and so on. So overall, OSIS oversee both of the clinical and analytical components of the in vivo BAB studies conducted. For the remote regulatory assessments that OSIS conducted, it's a virtual interaction between OSIS and the sites. So as we know that OSIS implement RA start from June 2020, right after pandemic started. In July 2022, a draft guidance was published, and as Julia mentioned, last month, an updated draft guidance also published. So there are several characteristics of the RAs conducted by OSIS. First is voluntarily participation. If the site agreed to participate in RA voluntarily, usually OSIS reviewer will request all the documents, records, or information being submitted prior to the virtual interaction. If there are some observations being, usually we call objectionable conditions, being observed during the RAs, those observations will be emailed to the site at the closeout meeting. So for the RA activities, we'll also provide the data reliability and subject safety recommendation to the review divisions. So RA activities actually allow OSIS to continue to oversee the conduct of the BAB studies, GLP, and animal route studies during and beyond the pandemic, which is support CDER review for NDAs, BLAs, INDAs, etc., and also support FDA's mission to protect public health. So here I'm just briefly introduce the RA process and inspection. For the RA preparation, OSIS reviewers will review all the documents and information submitted to FDA during the preparation. Meanwhile, we will send an email to the site, ask for voluntary participation. If the site agree to participate voluntarily, usually we just ask the site to provide all the documents prior the virtual interaction. Currently, FDA use the cloud filing sharing solution, which is box.com. For the inspection preparation, very similar, OSIS reviewer will review all the documents and information have been submitted to FDA. Opening meeting. Because RA is virtual interaction, so OSIS reviewer will schedule the meeting with the site through Zoom. For the inspection, it is in person. So OSIS reviewer will arrive at the site, present the credentials to the site. If it's a domestic, then FDA Form 482 will also be presented, and then have the opening meeting on site. For the facility tour, RA, usually we would like to ask the site to give us a virtual tour, so they can use any type of technology device. For example, any smartphones or smart tablets or camera. So they are going to show us the facilities in the Zoom meeting. For the inspection, then the facility tour is in person. OSIS reviewers will follow the site management or staff to visit the whole facility. Daily meeting. For RAs, usually OSIS reviewer will schedule 2 to 3 hours daily meeting with the site. So in that Zoom meeting, we will discuss any questions or concerns with the site management or staff. For inspection, the OSIS reviewers will stay in the site, review all the documents, as well as have the meeting with the site management and the staff. Closeout meeting. So if there are some observations in the RA, those observations will be written in email and sent to the site. But for RAs, we do not issue FDA Form 483. And for the closeout meeting of inspection, if there are some observations, then FDA Form 483 will be issued. So we can see from the side-by-side comparison of these two processes, there are some difference, but there are still lots of similarities between those two processes. Here's the studies that OSIS have conducted for the RAs. we do have lots of in vivo BAB studies, PK studies, ligand binding assays, immunogenicity studies that we conducted through RAs. And you can see we have even more variety of the in vitro BE studies that have been conducted during the RA. So all the activities actually is to support the regulatory decision regarding the drug application safety, efficacy, and labeling. So here I'm going to briefly talk 2 case studies that we conducted during the RAs. They are more focused on the analytical components of the RAs. So for the first case, here I just put what we observed during the RA. That OSIS observed that the site did not store the reserve samples for the reference product in an area segregated from the area where the in vitro BE study was conducted. So the story is that during the RA, after the opening meeting, we had a virtual tour. So when the site passing the in vitro study sample preparation and processing bench, we saw a small refrigerator standing just next to the bench. And then the site explained that refrigerator was used to store some chemical reagents and the reserve samples for reference product. So we did ask the site management to open that refrigerator for us. And then we did see in the upper shelf, there are some chemical reagents stored there. And in the lower shelf, the reserve sample of the reference product was stored there. And the interesting thing is that the site did have the reserve sample of test drug product stored in a designated refrigerator, which is located in a designated storage room. But for some reason, the reserve sample for the reference product stored in that small refrigerator next to the study bench. So we do collect the evidence that, and as well as the photos when the site submitted to us, we do collect the evidence that those reserve sample for reference product was stored not appropriately. But in the meanwhile, we audit lots of other documents, like, for example, drug accountability documents, shipment receipt, and we also audit the method validation study report. And for us, we see those, even though the site did have a regulatory violation, but not appropriately store the reserve sample for the reference product. But that misplaced of the reserve sample did not impact the study data generated from the site. So for us, our recommendation to the review division is that the data are still reliable, generated by the site. So when we give our recommendation to the review division, that will allow the review division continue the review. The second case study is a pivotal in vivo PK studies to support a BLA. So during the RA, OSIS finds that there are about 36 samples drug concentration documented as higher than LLOQ in the source data, but they are documented as lower than LLOQ in the submission to FDA. So we observe this discrepancy, and then we alert the review division, say, hey, there is a discrepancy between the source data as well as the submission. So that will allow the review division to send the information request to the sponsor and also allow the sponsor to give the explanation. So after that, the review division decides that explanation is appropriate and acceptable, and that little discrepancy did not impact the final PK analysis. So our RA recommendation also allow the review division to continue their review for that BLA. For the future, what we are looking for. OSIS will continue to improve and refine RA for its remote process and effectiveness. And RA is a critical and valuable tool that OSIS will continue to use to assist FDA in accomplishing its mission of ensuring public health and safety. Last but not least, the data submitted to FDA should be complete, accurate, and reliable to ensure safety, efficacy, and quality of drug products. Thank you very much. Okay. So I'll give this platform to Michael. Thank you.
I'm going to apologise for making this noise. Oh, that wasn't bad. Good morning, everyone. To those joining us online, good afternoon, good night. And that is the highlight of this talk. So Sean said, my name is Michael McGuinness. I am joining you from the UK MHRA. I have been well warned to slow down, and that will happen during the two presentations that you have from me today. I'm going to give you the MHRA's perspective on remote and hybrid inspections, to build on some of the sessions that you would have heard from Dr. Seller, Jennifer Adams, and Rachel Mead from the MHRA yesterday. What I'm going to do is talk about a specific bioequivalence slant, talk about some of the differences, and then provide some tips, logistics, feedback around the development. So without too much delay, we'll give you an overview of the bioequivalence program at the MHRA, talk about who conducts the inspections. We'll go through the inspection types. There is a small amount of audience participation. We'll talk about when the MHRA may make use of these types of inspections, and then we'll give you those, hopefully, hints, tips, things to think about. So, in terms of the inspection team, and contrary to what you'll have seen, is the MHRA bioequivalence inspections are performed by the laboratories and GLP team. The team that I head up, that is a multi-GXP team. So we have, as the name suggests, GLP inspectors, but also those that conduct GMP, quality control laboratory analysis, inspections, GCP, and as you would expect, bioequivalence and biosimilar inspections. We do have invaluable support by our GXP colleagues. Some of them are joining us today, joining from the GCP team. On the right, you can see my colleagues who obviously can't be with us today. We will hear from Emma Whale later on, and just to the development of the MHRA's hybrid inspection program, Emma was crucial to that. So I want to just acknowledge that. And Emma, if you're watching, hello. The one thing that, again, just to mirror what we talked about with Dr. O, is the MHRA's inspection program is both by the clinical element and also the bioanalytical element coming together. Now, you will see this slide again, but there are a couple of different elements I want to pull out. So you will see on the right-hand side the deficiencies that we have raised and pulled together for metrics between 2019 and 2003. But we'll come to that in the next session. We've conducted 24 bioequivalence inspections since 2019. Now, obviously, there is the downturn from the pandemic where we adapted. That also doesn't include any of the UK inspections that may be classed as bioequivalence, but would be considered a GCP laboratory inspection or a straight GCP inspection. And then the date that you can see there, so remote or office-based inspections were conducted between October 2020. We weren't quite as quick as the FDA. And that ended in June 2022. The reason for the delay we will allude to as we go through the presentation, but we recognise the crucial role in data review and weren't willing to compromise the outcome of the inspection until we had the correct tools. So, audience participation. I apologise to those online. We are going to talk about each inspection type as we go through, but for those in the room, I imagine if we asked you who had done an on-site inspection or who has hosted an on-site inspection, hopefully many hands, has anyone been through a remote inspection? Okay. Anyone been through a hybrid inspection? And then keep your hand raised if it was a good inspection. The FDA aren't watching. So yeah, we'll take these each in turn and talk about the development at the UK MHRA and how we brought that about. So this slide I call an inspection in a nutshell. Across the top in the lovely red, orange and blue, you will see the inspection flow. It's very deliberate that you will see that on every of the subsequent 3 slides. So you can see that the remote, hybrid and on-site inspections will follow the same process. So if we take it in turn, the notification and data requests, what we imagine is when that email lands in your inbox, there's a calm, considered response to what you're going to expect. At that point, it doesn't feel any different. You should know who your inspectors are, you will know the dates, you will have some information about what we intend to do, and also there'll be a long or short list depending on the scope of the inspection of requested information. We move into the inspection, which we will talk about, and then report and closure. The point to note here is that the MHRA don't distinguish between remote, hybrid, or on-site. We will produce an inspection report, and in GCP, you will get a statement of inspection detailing how it went, whether there were criticals, majors, or anything else. The part below that is a very high-level review of what we consider to be a labs or a bioequivalence inspection. So at the centre, we've got 4 big buckets, if you like. So we have the facility review, classically called the tour. You'll see that is the smallest of the 4 components before we move into interviews, discussions. Then we go up to systems reviews, so looking at quality management systems, quality assurance, quality control, document control, the list goes on and on. But the largest component, and we'll talk about this in a second, is for data review. Crucial to the bioequivalence inspection conduct is our ability to verify and reconstruct the information that you have generated during the conduct of the trial. We have heard from colleagues from GCP about real-world data, decentralized trials. A bioequivalence study, in its essence, is not that complex. It's usually open label, 2-product, crossover, and done in a relatively short period of time. The data that you generate during it, whether that's the clinical or bioanalytical, is crucial to our assessment when we're looking at it. So, the trained observers amongst you will see that my title says hybrid and remote, but we're talking about on-site. The reason I've put this here is just to enforce and highlight the development of the tool. You will see that, as you imagine, all activity is performed on-site. But what we've done in moving through the pandemic is realised that it is worthwhile us taking use and investing in our staff in terms of training, but also bringing in the analytical data systems. We won't go through them here because I can't endorse any of them. But we hold a number of software that are prevalent within bioequivalence studies. We request that data through Microsoft Teams, and we work within our own systems. This is a couple of highlights on our side. One, the training, it means we don't need to bother you on site. It helps with license management from your side, and then means that we can share in real time, but allows the development of this process. Remote inspections, as you can imagine, all activity will be performed remotely. So this was from October 2020. What we did here, and to highlight, is exactly as Dr. O talked about, is the use of visual technology to bring back that facility tour or a facility walkthrough. Now, we've trialed quite a lot of systems. We've used Microsoft Teams, Zoom, the list goes on and on. We've even made use of some augmented reality to test whether or not that would be worthwhile. it was mostly dizzy. But in terms of the remote inspections, again, it should feel like an inspection. You will see the boxes will definitely take longer. Referencing Jennifer's slides from earlier this week, an on-site inspection, the actual conduct generally is shorter, not factoring in travel. But where you start to be into remote inspections, and we'll come to it as we go to the tips, that element takes longer to pull together. What would normally be put a lab coat on, straight through, find the stairs, go there, turns into, right, we have someone that is possibly on the right side of the air shower, someone that is ready to go, multiple cameras. And those are a lot of hurdles that delayed our deployment of the remote inspections. Then moving into hybrid. So as again, you can imagine a hybrid is a cross between a remote and an on-site. These are not mutually exclusive, and the one thing to say is that during the pandemic, and I hope none of you have done this, but if at any point there was a reason to turn a remote inspection into hybrid inspection, that option is open to us. So we can escalate, so you go from remote to hybrid, potentially, and it brings the on-site component to it. One example was, as soon as we mentioned the word critical finding, the camera went off, the microphone didn't. And it was interesting. So the hybrid inspection, again, pulls together all of those components. So we are working within Microsoft Teams. We'll invite nominated colleagues from your sites into our Active Directory. We will deal with all the communication. We will request documents. We will host meetings with you, provide constant feedback. We're there on Instant Messenger if you need us. Video calls, tours. And it should, again, feel like a real inspection. There are segregation of activities, so in the nutshell part, you can imagine the facility walkthrough will be done on site. The systems review will be split. Data review will be a large part done remotely in the United Kingdom before we turn up on site. So the idea is that we would run through all of the analytical data and turn up with questions, therefore making best use of your time and ours. Interviews, again, possibly split. So, when will the different types be used? And again, this is dependent on several factors. The first one is compliance and inspection history of the site. So we take each component in turn. The compliance, whether or not that was good, bad, or indifferent, will determine whether we're there. So if we're looking to confirm closure of a critical finding and that related to a facilities and equipment issue, it makes sense to be on site. If that related to a software validation issue, which we could bottom out prior, then you might see us as a hybrid or remote inspection. The inspection history, the point to note here is we will predominantly, if you are the first time you've had an inspection from the MHRA, we want to see what the facility looks like, walk through, for want of a better term, make sure all the lab coats are in order, and then continue there. So you are likely to see an on-site inspection. The nature of the inspection is the second part, so whether it's routine or triggered. So the MHRA conducts systems-style inspections for, if you like, frequent flyers of bioequivalence generating sites. Whereas if it's triggered, looking at a specific issue, what we might want to make use of is the effective use of your time and our time. So possibly lends itself to an on-site inspection. And then the last one, and we'll talk about this as part of the review, but the suitability of the inspection site. If your Wi-Fi is so terrible that you can't download a PDF, it is unlikely that you will be able to support our Microsoft Teams video tour. So we will ask those questions up front. We possibly switch to a hybrid inspection after that notification, and then we move forward. So this section is related to genuine feedback that we've received as part of the development of the tool. And as we continue to do this, I would encourage you, if you are subject to a remote MHRA inspection, a hybrid MHRA inspection, please do continue that feedback loop. So the use of visual technology. This one is crucial as we work through, but ensure sufficient connectivity through the facility, whether that's through Wi-Fi, 4G. What typically happens is when you approach an LCMS, all of the systems go down. It doesn't like the interference. Consider the movement between areas. It's very easy just to consider that an air shower is normal, a water shower is very difficult for a computer, for a phone or a camera to go through. So yeah, bear that in mind. Possibly multiple devices is what we've seen people do. Time zone challenges. So as I mentioned, the bioequivalence inspections we're talking about in our metric here are our overseas inspections conducted, whether that's in India, whether that's in the US, whether that's in Canada, to name a few. But the UK obviously is not in that time zone. So you may find that you can see us for part of your normal working day. So again, discuss this as part of your planning process with your lead inspector. We are used to this and well practiced. It may mean that we have 4 hours with you, and then when that goes down, we will work on our own, submit document requests, and then arrange a meeting to discuss that with you the next morning. There is not an expectation for you to work through the night, nor turn documents around at your 12 o'clock. So agree with colleagues when they will be available to support the inspection. Again, your lead inspector will walk you through this. The second part is in providing the analytical data. So one of the things that we learned is that everybody's experience with analytical data varies. How you can upload it, whether or not you zip it, all of that we have walked through. So we have come up with a guide for the majority of the systems that we have, and that will be provided to you to try and take some of the thought process out. So if you follow that, you should be all right. If you are encountering any challenges, please let us know. It's a crucial part of the inspection for us. And we have found on occasion where we've started the inspection, think that we've got everything, and then look, lo and behold, all the WIF and injection files are missing. And what that does is it raises the inspector's blood pressure and you start off on the wrong foot. So avoid that. Do share. So in inspection progress, what we'll do is we will try to arrange this as much as possible, but agree feedback sessions. As we said, depending on your time zone, that might be better the day after. We will share our concerns. If there is anything we perceive to be a deficiency, certainly at the critical or major end, then we'll do that there. We'll give you time to understand our thinking, potentially provide a rationale for why that might be the case, and then we move forward. Inspectors should share feedback as they go through. In terms of the last one, so during the inspection, we've talked about a number of systems, so whether that's Microsoft Teams, Active Directories, whether that's the analytical data, if the use of a system or process is causing you a hindrance or administratively burdensome, let us know. It's not designed to be a stress test for you. Most of us have been on the other side and received the exact same treatment. So we are trying to get as full an understanding of your systems, your procedures, in order to make a relevant compliance assessment to our assessors. It is not designed to put you through the ringer. So come back to us, liaise with your lead inspector, and provide that feedback to us. There are a number of avenues open to us to change document requests, to switch systems within reason. If you tell us you don't want to give us your analytical data, that's going to be a red flag. So in summary, we've embedded the new tools during the pandemic into routine processes. You can see that with the on-site inspections. That analytical data upload took us a long time to put right. It is something that we have worked hard on and will continue to improve and expand the number of systems that we have. But it's definitely been a big plus for us. We continue to look for new ways to improve and our remote and hybrid inspections. I do have a Disney quote, but I'll come back to that. Your feedback on the inspection experience is, as always, welcomed. As with anything, the MHRA prides itself on its transparency and pragmatism. We talk about it during our training and accreditation, and it's something that we would encourage. There is no stupid question during an inspection, within reason. And then the last one, before I give you the quote, is the aim of the inspection, regardless of the type, remains the same. We want to understand your systems, your procedures, your quality of data, your protection of the subject, it's the study participant, and the well-being. And that, before I finish, is the quote that I have for you is, find the right tool and you'll make magic happen, from Toy Story, where Woody tries to encourage teamwork. And with that, Sean, I'll pass back to you.
Thank you so much, May and Michael. Really appreciate your presentations this morning. Now, for our next session, we are taking a focused look at the inspections of analytical conduct of bioequivalence studies. So in addition to another presentation from Michael, which you all get to look forward to, we are also pleased to introduce another essential member of OSIS, Dr. Igwe Cynthia Zhang. Dr. Zhang is a senior staff fellow in OSIS. She plays the integral parts of conducting, coordinating, and reviewing comprehensive FDA BIMO clinical, analytical BE inspections, as well as supporting our GLP team. So please join me in welcoming Cynthia this morning to speak on bioanalytical issues. Cynthia.
Thank you, Sean, for the kind introduction. Good morning to everyone in the room, and good afternoon, good evening to the online audience. I am thrilled to be here today to talk about some bioanalytical issues found from the recent FDA BIMO inspections and remote regulatory assessments, RA. Here is my FDA disclaimer. The learning objectives of my talk today include to get familiar with the relevant US laws and regulations, to understand the basis for demonstrating bioequivalence, BE, and also to make you aware of some bioanalytical issues from recent FDA BIMO inspections and RAs. And here is the outline of my talk today. In 1984, the Hatch-Waxman amendments of the Federal Food, Drug, and Cosmetic Act introduced the new abbreviated approval pathways of marketing applications. Specifically, a 505(b)(2) application is an NDA containing full reports of investigations of safety and effectiveness, where at least some of the required information comes from studies not conducted by or for the applicant, and for which the applicant has not obtained the right of reference or use. Meanwhile, a 505J application is an ANDA for a drug product that is a duplicate of a previously approved drug product, and generally must contain information to show that the proposed generic product is pharmaceutical equivalent and bioequivalent to the reference listed drug, RLD. Additionally, the Code of Federal Regulations, CFR, Title 21, also includes relevant sections. For example, I listed here, there are Part 312, the Investigation on New Drug Application, Part 314.54, Part 314.92, as well as Part 320, Bioavailability and Bioequivalence. Here, I am showing you all the table of content for CFR 21, Part 320, Bioavailability and Bioequivalence, which lays out the legal foundations for requirements of bioavailability and bioequivalence studies. You may find the details online if interested. In Part 320, the basis of demonstrating BE is defined. Specifically, two drug products, test and reference listed drugs, RLD, are considered as bioequivalent if they are pharmaceutical equivalent or pharmaceutical alternative. And the rate and extent of absorption do not show a significant difference when administered at the same molar dose of the active moiety under similar experimental conditions, either single dose or multiple dose. And if the rate of absorption is different, the difference would be intentional and reflected in labeling. And the extent of absorption should not show a significant difference between the test and the reference, or the RLD. For drug products that are not intended to be absorbed into the bloodstream, BE may be demonstrated by using scientifically valid methods that are expected to detect a significant difference between the test and the reference products in safety and therapeutic effect. In addition, FDA also publishes guidance documents to describe the agency's current thinking on certain topics. The guidances do not establish legally enforceable responsibilities and should be reviewed only as recommendations. For example, here I'm showing you the guidance M10 Bioanalytical Method Validation and Study Sample Analysis published in November of 2022. This guidance describes recommendations of the bioanalytical methods used to measure concentration of chemical and/or biological drugs in biological matrices obtained in all phases of clinical trials in regulatory submissions, including comparative BA or BE studies. And the table of content displayed here is showing some expected critical elements during method validation, including selectivity, specificity, matrix effect, calibration curve and range, accuracy and precision, carryover, dilution integrity, and stability. Last but not least, the FDA also has the Bioresearch Monitoring Compliance Program, BIMO. It's a comprehensive program of on-site inspections and data audits designed to monitor all aspects of the conduct and the reporting of FDA-regulated research. The BIMO was established to assure the quality and integrity of data submitted to the agency, FDA, in support of marketing applications, as well as to provide protections of the rights and the welfare of human subjects and animals involved in FDA research, FDA-regulated research. Among several BIMO compliance programs, the program 7348004 describes the coverage of a typical in vivo BABE analytical inspection, like I'm showing you here. Again, you may find it online if you're interested for more details. Now, let's switch gear, and I'm going to talk a few case studies. The first case study, this one, is a BE study with PK endpoint submitted under ANDA. The drug product was a small molecule and also an endogenous compound. The study was designed as a randomized two-treatment, two-sequence, 4-period, crossover, single-dose, and full replicate trial. And the bioanalysis of the samples was conducted using LC-MS. During the FDA inspection or RA, objectionable conditions were observed. Specifically, there were higher pre-dose analyte concentrations observed in the subjects that's receiving test products compared to the reference listed drugs. Not all the subjects, but only some of them. And also the pre-dose analyte concentrations did not exhibit expected physiological variability in the subjects. The firm being inspected could not provide adequate explanation or justification of the above observation. No discrepancy was identified during sample analysis. However, like I mentioned before, the firm could not offer any explanation or justification would support the observed pre-dose discrepancies between the test and the reference products in this subsets of the subjects. Therefore, OSIS concluded that the analytical from the clinical study are not reliable for agency review. Case study number 2. This was an in vivo BA study with PK endpoint submitted under an NDA. The product was a large molecule and was analyzed using ligand binding assay, LBA. During the inspection or the RA, objectionable conditions were observed. Specifically, the assay actually did not demonstrate sufficient accuracy to measure the analyte concentrations. Specifically, the calibration standards and quality control samples, they were not prepared using a reference standards of known purity. Nonetheless, the calibration standards were used to back calculate, assign, and report specific analyte concentration for the subject samples. This This subject's concentrations were later on used to evaluate and support the endpoint in the clinical studies. OSIS concluded that this observation impacts the study data reliability because the site did not use analyte reference material of known purity to prepare the calibration standards and QC samples. Thus, it is impossible to calculate and report accurate analyte concentration of the study subject samples. However, the percent change from baseline analyte data should be reliable because the method actually demonstrated adequate precision when measuring instrument signals from the QC samples. That is, the method consistently measured higher signals in high QC samples and low signals in low QC samples with adequate reproducibility. OSIS inspectors also verified that the instrument was properly maintained and calibrated according to the firm's SOP and CLIA requirements. Thus, the method showed adequate precision and the percent change from the baseline analyte data were reliable. All right, now it's time for some challenge questions. Challenge question number 1, true or false? You must follow all the recommendations listed in the FDA's guidance for industry for bioanalytical method validation and sample analysis. A, true, B, false. Can I get a show of hands? Who's picking A, true? Half, maybe 0. Okay. Option B, oh, sorry, I gave up. Yeah, so the answer is false. Like I mentioned before, earlier in my talk, the FDA guidance documents, they are not legally enforceable and they should be viewed as the recommendations only, and it reflects the agency's current thinking on certain topics. All right, challenge question number 2. Which type of evidence may be used to establish bioequivalence BE? A, comparative PK study. B, comparative PD. C, comparative clinical endpoint. D, in vitro in vivo correlation, IVIVC. Or E, all of the above. Can you guys just shout out what you would choose? Perfect. So all those about 4 study types could be used as evidence to demonstrate bioequivalence. And here are some references that I mentioned during my talk. And if you are interested, you should be able to find them on the FDA websites. With that, I would like to thank my OSIS colleagues and management support to make this presentation possible. Thank you so much for your attention, and I'm happy to answer any questions you may have during the panel discussion. Thank you. All right, now let's welcome Michael to the stage, and he's going to talk about MHRA's perspective in their findings from recent inspections. Welcome, Michael.
Hello again. So as Cynthia said, I'm going to take a UK slant and talk about bioanalytical observations and findings from recent inspections. Again, just to bring this slide back up, we'll talk about something different. So concentrating on the very well put together graph, 3D image you can see on your right, distribution of findings. We'll go through not in a lot of detail, and you will pick up some of the clinical ones from Emma's presentation later on. But there you can see the number of critical findings, major findings, and other findings that we've raised since 2019. The aim of this presentation is slightly different. We have a slightly mixed audience. We've got pharmacovigilance later. We've got bioequivalence in the morning, both clinical and analytical. So the findings that we talk about here have been deliberately selected to try and give you food for thought, and with the idea in mind that if you take care of these, you won't end up in the critical finding list. Just in terms of, I said there'll be 2 audience participations, but one of them includes a prize. For this one, by show of hands, can I ask those that are directly connected with regulated bioanalysis in the room? If you're online, please feel free to raise your hand. I just can't see you. So anyone directly connected with regulatory bioanalysis? Hannah, thank you for your hand. Okay, that's great. There is no problem. We will walk through all of these together. So as I said, here's a breakdown. You will find this again in Emma's slides. So in the red or orange box, depending on what you can see, that is what we've classed as our clinical findings. And Emma Whale will go through that in session number 3 later today. What we are going to talk about is the major findings in the blue box. So as you can see there, method validation, data integrity controls, and clinical sample analysis. As I said, the intention here is to stop those findings turning into these findings. It has been very deliberately ordered that you can see method validation, data integrity, which is the, if you like, big brother or sister of data integrity controls, and clinical sample analysis. So as I said, we've got 3 examples to talk you through. Just in terms to give those context, some of you may have experienced an MHRA inspection in the past. We classify our findings in those categories. So a critical finding is where a significant departure from the principles of GCP have been identified, which either put the subject, clinical trial subject's well-being at risk, or the integrity of the data cannot be relied upon. A major is something that, if action is not taken immediately, will result in a critical finding. So if you like, it's a warning shot. So just to take that down, we'll break this into the method validation example I've selected is around stability. The clinical sample analysis will be on run acceptance, so how you decide whether or not an analytical run is valid. And in terms of data integrity controls, we are going to talk about dynamic data. So what I want you to think about as we go through these examples is making effective use of regulatory guidance. Cynthia's talked about that. Guidance is exactly there. It is not a thou shall, but if you're going to do something different, you need to think about it, document it, and go through. The idea of transparency and how crucial that is, particularly where you're going to do something innovative or novel. We want to be able to reconstruct your thinking, your processes. We don't want to rely on your colleague's memory when you've won the lottery. And the last part there is data security and control. It is unfortunate that a number of inspections in my 9 years at the agency have resulted in negative outcomes, not necessarily because the method doesn't work, not necessarily because they weren't diligent, but because the system which you had acquired the information to leaked information. It was open to abuse, and it's one of the things that is incredibly frustrating as an inspector. So, to go into method validation, on the picture you can see there is a lovely -80 freezer with some samples. What I want to think about is long-term stability is the assessment of stressing the samples by putting them in to a freezer designed to mimic subject samples during the clinical trial and pulling them out at defined points. If you think about it like a lottery ticket and waiting for the draw, you put them in hope. And then as each number comes up during the draw, you get a little bit closer to the jackpot. You get to design your subject samples. The bit I want to talk about, and what we've seen on inspection, is that a number of laboratories have failed to analyse the prepared samples, stability samples, immediately after generation. So if you like, you're your lab scientist, you've got your lab coat on, you've got all of your samples in front, you are spiking your low quality control samples and high quality control samples ready to freeze. We'll go to the guidance part in a second, but at that stage, you have a huge amount of variables that can affect the outcome of the assay. Whether the analyst's pipetting technique, if that's what you're doing, is on point that day, whether the tip has got a manufacturer's defect, whether or not there is something else at play within the method. The idea of doing the analysis up front is to give you as clear an indication of what you can see later on, that you knew it was all right when it left there. Now, again, I'm going to share some ICHM 10 references, and Cynthia's links are there because I tried to hyperlink this and it didn't work. So what I've done is taken the direct wording from ICHM 10. So this is specifically section 3.2.8, handily titled stability. And the part in bold is unambiguous. It talks about aliquots of low and high QCs are analysed at time zero. Going back to my hamstrung lottery example, it is the one thing that you can do to improve your chances of the jackpot. It gives you future information about it was okay at the time. And it's a missed example, sorry, it's a missed opportunity if you don't take it. So, what we'll do there, the text you can see currently is a slimmed down version of the deficiency. So laboratory procedures did not contain a requirement to analyse stability samples immediately following preparation. Therefore, there is no assurance that stability samples met acceptance criteria prior to storage. Now this has a number of outcomes as we work through it. But at this stage, long-term stability is something that you do right at the start. It's done in method validation so that you can lay your samples down because you have to wait for them to age. It is not something that if you get to the end of your clinical trial that you can jump back. You have lost that opportunity. So what can you do if this is your facility? And this is a very common finding that we're seeing currently. We've presented on it in the past, and I'm hoping in talking about it today, I can stop writing that finding. So the first thing is to recognise that you have a problem. Look at whether or not you have time zero samples. And I will find most of you that work with laboratory scientists, they are quite thrifty. They will turn one experimental procedure into many. So you might find, although it wasn't designed to be time zero, that your low and high QCs might actually have been analysed by luck. And some of the cases we find on inspection, that's exactly what's happened. So that's the first case scenario, what I would call the best outcome. The next part is if you don't have that, then you're starting to look at the reliability of the long-term stability, how it's deteriorating over time, the percentage of nominal for your low and high QCs. And that's a really difficult thing to do because what you're doing is replacing an experiment with a human's opinion. And as all inspectors will tell you, we would much rather have data. Now, it's not the end there though. You might have seen that your time point 1 to time point 5 all come out beautifully in line with nominal, plus or minus 6%. So well within acceptance criteria. But you might also find that time point 1 passed and time point 3 passed, but time point 2 didn't. And you're left in that horrible situation where you can't say it was okay when it went in. And that situation crops up more than you would think. So again, try to incorporate this into your procedures as early as possible. If you don't have it, it's not the end of the world, but you should start documenting that now. The next one we're going to talk about is clinical sample analysis. And again, this is run acceptance criteria. So I know a number of people in the room don't work directly in regulated bioanalysis, but this is the part when you get to the end of the assay, you've got your calibration curve, you've got your QCs, and what you're doing is looking to see if you can trust the unknown concentrations that you've got as a result. So those are incredibly well designed. They're to take away any subjectivity or ambiguity that it shouldn't be my decision or your decision. It should be that's what we've agreed up front and that's what we're doing. So what are we seeing on inspection? The inclusion of company-specific criteria. So we've mentioned ICH M10, FDA have their guidance as well. In addition to that, previous to that, you have the European Medicines Agency's Bioanalytical Method Validation Guidance 2012. And what those talk about is a defined list. So you're looking for assay accuracy precision, you're looking specifically at the calibration curve, whether or not you can regress lines against it, you're looking at your extraction method through QCs, you're gathering a wealth of information and then putting that back in. So that's one thing. If you as a company decide we want the best data, we want our gold-plated analytical results that are irrefutable to a regulator, and we're going to insist on this. That is, as we've said, a possibility. Document it in your SOPs. The second part in bullet point 2 is that these additional criteria are not always applied consistently. This is where you end up in a problem with the inspector. Because as we said, bioequivalents are slightly different to some trials. The design is relatively simple, 2-period crossover, open label. The outcome for a comparative PK assay is very well defined. You know what a pass-fail is. Most regulators, all regulators, I would say, will be able to tell you that, a confidence interval of 85 to 110, or 90 to 110. So now you've got this real fear that bias, whether it's conscious or unconscious, can be introduced by the decisions that you make So something that would normally have been regulatory guidance, unambiguous acceptance criteria, suddenly becomes, that's just an SOP deviation. And that's the dangerous place to put an analyst or a scientist in, which might mean that they have to work a double shift at the weekend. So take the subjectivity out. What I would say, if we go into, again, it wouldn't be a regulator's presentation without where we're taking this from. So I've slimmed this down from ICH M10. But again, it's really clear. It talks about rejected Cal and QC calibration curve. This is a specific example for a comparative PK assay, where the company were including a criteria. If I go back. Nope, apologies. Where the company had included their own criteria that if they lost the lower or the higher calibration curve, then the run would be rejected. Regulatory guidance incorporates the reason why you might not want to do that, so long as your QCs are covered within that run. And for the example that we're talking about here, we saw a selective use of this criteria being applied. So, again, on the right-hand side, from there you can see the deficiency that we would raise. So laboratory procedures were found to contain issues with respect to analytical run acceptance criteria, where requirements were not in accordance with guidance. Consequently, and this is the part where we're talking about bias, the policy for analytical run acceptance would have otherwise been, as a consequence, the policy for analytical runs, which would have otherwise been considered acceptable, were repeated, and the original analysis not considered for pharmacokinetic output at the end of the trial. Now again, in terms of considerations, one thing that I would challenge, whether you're working with a bioanalytical lab, you're a study monitor, is one, are these additions essential? Are we talking about gold plating, or is there a scientific reason for why this needs to be in your comparative BE study? And if they can't answer that question, my suggestion is that you ask them to work to the guidance. You take away any form of guidance. They might have a very well-documented rationale. Again, when we've seen this, where we've seen that it being applied inconsistently, again, one thing that we've struggled with is that documented rationale. Why are you doing it? Why is it in place? What can be done? Okay. So, the last example that I want to talk about is data integrity controls, which seems like I haven't stopped talking about it since 2018, and the use of dynamic data. So what are we seeing? And again, this is in the major category. We have examples where this has failed and we're talking about criticals, but this is the precursor to there. This is where you have the opportunity to affect change. So what are we seeing on inspection? We're continuing to see static data being used for review of bioanalytical data, which has been acquired in a dynamic format. Just skipping ahead to the MHRA's guidance and a plug, other guidances are available, including the FDA's. Raw data must permit full reconstruction of the activities. Where this has been captured in a dynamic state and generated electronically, paper copies cannot be considered as raw data. What I'm going to do is take you through an example, and this is where the prize and audience participation comes in. What we've got here in the picture is a chromatogram, and it's been used for stability assessment. It is generated in a system with well-defined and what I would call broadly GCP and GXP data integrity controls intact. If we go slightly closer, actually, let's do audience participation. This study was from a comparative PK study where you had LCMS, MS, but not to mislead you, but this chromatogram better shows my example, but it is not from an LCMS, MS. Anyone in the room want to offer a potential, what method this might have been from, what machine? And I can offer you a carbonated Scottish soft drink. Feel free to shout out in the room. Again, online, I'm really sorry. Not LCMS, MS, anything else that would be in that chromatogram generating sphere before I open it up to my colleagues on the right-hand side. Okay, that is fine. Anyone from the inspectorate, whether UK, FDA or Health Canada, want to offer? No, I'm running ahead of time. No. Other soft drinks are available. Okay, so this again, we're talking about dynamic data. What this company were doing was printing out the chromatograms and presenting it for review. Now, Again, everybody in the room, I imagine, has some experience in quality assurance. There's something that sticks out about this, something that doesn't appear right. That is a lovely gap down the bottom. So the chromatogram printout was modified prior to the review stage. And what I would say here is the removal of the acquisition date and time sticks out like a sore thumb when you highlight it. Now, the reason this is a problem is we're talking about stability. It's not the chromatogram itself. The chromatogram actually was acquired very well. It was within acceptance criteria. It can be used. But the timing, the metadata when you acquired that injection is incredibly important, because what it does is show that I can rely on your data. And what they'd done is forgot to take the sample at the right time. So the scientist, in some misguided attempt, said that's fine. And they waited that out and submitted it to QA and QC, like all the way through until we turned up on inspection. And we are used to looking at gaps, so we found this. So in the interest of time, I'm not going to read the deficiency. But again, it's there. You can see that the dynamic state is crucial to it. One, you've spent time, money, generating a system that can generate all of your analytical data with audit trails. It's got a Watchman directory that moves it out of a modifiable format. You might have Active Directory to link it directly to an individual. There is no way of changing it. But yet you print it out and you let them submit it. So in terms of considerations for this, I want you to think about why would that be necessary. One, it's easier. I can take a piece of paper, I can go and sit at my desk. It takes a bit more thought to understand why you might want to then go to the system, go to the lab, have the yellow light, have the noise. That's not a conducive environment for review, but it's not the reason to not do it. The removal of the opportunities is only part of the puzzle. And I think I've gone through 3 examples, and what I would say is, if you remember nothing else, it's the second part of this. Creating a system that is tightly secured and tracked by audit trails, the modifiable data is moved by Watchman directory to a safe, secure location, the archive is all intact as one piece. Humans are relatively resourceful. And again, in 9 years, I've found that people have found a way if they want to do this. So what you need to do is to achieve the right environment where they can make mistakes. Not doing the stability sample at the correct time is not the end of the world. Removing the acquisition date and time is. That is the one that breaks regulators' trust. We then talk about where else have the modifications come. And going back to that, that is a very difficult crack to fill. So I want to pull this up. The idea of the talk was to try and take you from that wiggly blue box to remove them completely and avoid getting to this grey-black box. What I want to leave you with is a short summary. Look at regulatory guidance. It is our current thinking. If you do have a good reason for doing something different, make sure you document it. Your decision-making and the documentation around that is crucial. We don't want to rely on hearsay when we turn up to do an inspection. Exercising control of your data is important. Don't let all of your hard work with the CSV, the periodic reviews go to waste. And act before issues escalate. And with that, I'd like to thank you for your attention and pass back to Sean.
Once again, a big thanks to Cynthia and Michael. We are now going into a break stage. We have 20 minutes, so aim to get back at 10:20. And for those online, appreciate your flexibility with the change in the agenda. And we'll have our question and answer session for the first 2 sessions at 10:20. See you in a second. Thanks. Thanks for everybody. Starting our session 1 and 2 discussion panel with our presenters. Again, thank you for everyone for submitting their questions, and we've got a good set to discuss. So jumping right into it, we have an interesting question about our India office. And so in the FDA has an office in India, and the question is, do FDA personnel in India conduct FDA inspections? So Maid, would you like to answer that?
So would you mind to say which question?
This is, we have the FDA India office.
Oh, okay. So before I answer the question, can I say the disclaimer is still usable right now? So all the answer is on my own, not represent FDA's policy. Okay, so if that works, so I would like to answer this question. So the question is, what is the purpose of FDA office in India, and does FDA personnel conduct FDA inspection in India? Yes, we do. FDA do have Indian office, so there are inspectors present on site when we conduct the inspection in India. Meanwhile, our OSIS reviewers also travel to India to conduct the inspection on site. Thank you.
Thank you.
And,
Mike, you wanted to add anything?
Yeah, the second part of the question talks about, does the MHRA have an office in India? No, we don't. Our inspectors travel from the UK to wherever the bioequivalence site is located.
Excellent, thank you. Okay, well, we have another question for, I think, for May from your presentation. Can you just clarify the differences between the RIE, the RR, and the RRA acronyms we've been
Yes, for this question, I will have to defer to FDA guidance because we understand there are several names have been used for remote. So guidance already have very clear description regarding those names and the function. Thank you.
All right. Now, here's a question I think, Cynthia, maybe you could take. A question about the performance of RRAs, and is it performed, what time within the BA/BE study conduct do the RRAs get performed?
Thanks, Sean. That's a good question. Usually, we would perform the RAs for inspections after the completion of the study, and the sponsor or applicant submitted the application to the FDA. However, during the RA or inspections, sometimes if the study was conducted, let's say 10, 15 years ago, we would also like to have a look of the firm's current practice. So we usually require or request like a master list of the BAB studies conducted during the past 5 years. And sometimes we would pick some more recent studies, sometimes even ongoing BAB studies to evaluate the firm's current general practice of bioanalysis. So I think that helps.
Thank you, Cynthia. Any additions from our other panelists? All right. Now, here's a question about remotes in general, and we've discussed how they're voluntary, but what happens if a site refuses? Sort of a question for everybody. Anyone have a thought to share there? What are the consequences?
I mean, I think if I start off from an MHRA perspective, obviously we will ask the reason that you're refusing. We consider remote, hybrid, and inspections to be inspections, on-site, sorry. And I think with this question, it's the same as anything else. Our reason for inspecting is there to review the data and ensure it's suitable for an application. It is unlikely to shorten the application's length. It's more likely to prolong it, but we would engage with you at that point.
All right. Thank you, Michael.
Yes, similar to FDA. So if we meet the refusal for the RAs, at that moment, maybe there is no consequence, but FDA still have the authority to issue the inspection. If inspection would not be happen, hypothetically, so the review process could be delayed. So I think that's the consequence we can think of. Thank you.
And I think another example I can think of is some of the sites where they are located is under impossible circumstances for them to host the RA. For example, some experiencing wars or there's some technical difficulties. So in that case, we understand, the difficulty, and we would try to do some remote data audit without the participation of those site staff.
All right, thank you very much. I think, the message of flexibility and the use of the remote tool really as needed is an important one. A question about observations and the inspection reports. Are observations included in inspection reports? I don't know, this is probably for everybody. Mike, do you want to?
So again, just from whether observations are included within inspection reports, yes. So whether it's hybrid, remote, we will incorporate everything into that inspection report and issue it to you, but that's our position. In terms of, we use observations in a slightly different way. So we have observations at the bottom, which may not necessarily be a deficiency. So just in case there's a terminology issue there, they might be things of note during the inspection, which are for your information.
Excellent, yeah, good distinction. Here's a good one that we hear quite a bit is what's the reason for not issuing 483s with an RRA?
I can answer that because RA is not an inspection. So 483, the full name is Notice of Inspectional Observations. So that is why we don't issue 483 for RAs. Thank you.
All right. Now, question about feedback. How do we, as regulators, or do we accept feedback from a site post a remote? And is there an evaluation form being provided to help sites provide guidance feedback?
Okay, I can answer that. So we do welcome feedback. Currently, every time when we finish the RAs, we provide the email box, which is cedar-osis-beq@fda.hhs.gov. So we welcome any feedback. And if you have any form you like to use, please also email to us. Thank you.
Yes, please.
So specifically in the idea of a form or template, the MHRA don't make use of one. The one thing I would stress is not to wait till the end of an inspection or a remote observation to raise anything. What your lead inspector should do is try and tweak the inspection to remove any burdensome conditions and work with you. And we do that in real time. So the feedback that I described during my first presentation was related to real-time feedback that we received during the inspection. Equally, we are fully contactable after the inspection, and we will take anything that you just didn't think of at the time but want to let us know.
Thank you. I think that's a really good point, that the more feedback we get, the better the tool can be developed over time. And so it's really important to have your thoughts shared as they come up. Now, regarding remotes and the discussion of virtual tours by video, is it required to have a live tour, or is it okay to have a photo tour or a pre-recorded tour? Anybody want to answer that one?
That's a great question. I would say that the live tour is highly recommended. Especially during the RA, you're welcome to upload the, photos or pre-recorded tour to box.com as you wish. But during the RA, we still hope to see your facility live, because a lot of things could have been changed since the recording of your tour or the photos being taken. And the live one would be, a better reflection of the current situation of your facility. And that will give us a better idea of how your facility operates. Thank you.
Excellent. Michael, you want to add?
Yep, just to say, in terms of, we do receive photo tours, what I would consider a business development video at the start. One of the key things for an inspection is the interaction between the inspection team and the inspectee. And a video tour doesn't allow us that chance for collaboration, discussion, and questions. So although you might provide it, we would always insist for a remote inspection that we do a live tour. If that proves cumbersome, then we might move to hybrid.
Excellent, excellent. Now, regarding, again, a question on remotes, and I know this came up in the past sessions yesterday, When do we see remotes being used in the future, past COVID?
I'll say when needed. So we'll still continue using RA as a tool past COVID. Thank you.
Excellent. Michael?
I think Dr. Sellers said it yesterday, whenever. And it's true, the remote inspection is a tool. If there's a reason that we want to take a specific look at something, we will make use of that. It's not restricted to COVID. We've spent a lot of time developing these tools, as the colleagues in FDA have. And if it's to answer a specific question, then that might be very well the best way and route to do that.
Excellent. Thank you. Let's see. Wrapping up a couple of there's a few more remote questions. One of them is, in terms of, written observations, and as you mentioned, written deficiencies really of a remote following an inspection. In terms of at least the FDA side of things, what's the status on classifications?
Again, because RA is not considering inspection currently, so FDA do not classify the observation. Thank you.
Thank you, May. And also, let's see, in terms of in conducting of a remote, what kind of information is asked for from the site?
For the BAB remote RAs conducted by OSIS, we really follow our own SOPs. Usually there will be communication letter 1 going out to the firm asking if they are willing and able. If they say yes, we will continuously sending out communication letter number 2, which would identify the studies. And we will also include attachment A, which is the detailed list of the requests of the documents. Usually those documents would include, I would say 2 to 3 parts. The first being the general administrative information, like the overview of your firm, your SOP index, your flow plan, your like the list of personnel involved in the studies and their training records, the list of equipment used and their calibration records. And then going on to the study-specific information, we would request for the all raw data. For example, if you use like a hard copy of the lab notebooks, we would like to see those, I don't know, either scanned copy or the electronic copy you already have. And then there would be, for example, the preparation sheets of your calibration standards and QCs, audit trails. And we may also asking for some electronic file, for example, the individual run, the results and acceptance criteria, and your sample repeat. Also the correspondence with the sponsor or clinical sites, if you're an analytical firm. And regarding sample receipt and the tracking, handling, chain of custody. In addition, if this is in vitro B study, there will be reserve sample requirements per 320 regulations. So we would asking for information related to the reserve samples for the IP, investigational products, and also the storage conditions, again, chain of custody. And any deviations you have documented or any investigation of failed runs, or justification of repeats. So just a few examples.
Excellent.
Yeah, but if you happen to be selected for RA, you will definitely see more. Thank you.
Thank you, Cynthia. Michael, do you want to add anything from UK's perspective?
The only thing to make a specific reference to, because I think Cynthia's list was very good, was just specifically on the analytical files we request. You will get notification of what that looks like. Traditionally, it's all analytical files associated with method validation, sample analysis, and audit trails that bridge the gap the month before, the month after. So month before validation and the month after sample analysis. But that could be subject to change.
Yes. And that actually leads into questions focused on session 2, is what are the specific software that MHRA holds and use for review of analytical data?
And this is as of the current time. We're currently looking to install and embed others, but the current list is SciEx Analysts, SciEx OS, Waters MassLinks, and Empower 3. That list is growing. We are looking at seeing what's the most prevalent software within industry. We're looking at adding LBA assays, for example. But yeah, as of this time, that's the current list. But it's fair to say that we have now worked through that process and can add to it.
Excellent, excellent. All right. Now, of course, something that's on everybody's mind is the use of AI. And so one of the questions that we got was, what is the use of AI in bioanalysis as we've seen it, and any thoughts on acceptable, unacceptable uses? Yeah.
So yeah, I can start with this one. In terms of have we seen it, the simple answer is not yet. We are expecting, if you imagine, you can automate certain aspects of whether that's bioanalysis, the run validations, etc., interpretations and integrations. But in terms of what is acceptable and unacceptable use, is we're still very much at the stage of treating it like any other computer system, and the applications that you would see there from a GCP point of view apply here. It's worthwhile mentioning that artificial intelligence is being used broadly as a catch-all term for machine learning, generative AI. So the functionality is key to what it's doing for you. And with anything else computer systems on inspection, we want you to demonstrate that it's fit for intended use, and you can demonstrate that through documentation.
Excellent. Well, I think that is a nice way to wrap up all of the questions that we've received. I want to appreciate and thank all of the panelists again. Thank you for your time and your presentations, and please join me in a little welcome for them. Thank you. Next up, we are going to move on to our BABE clinical session. I think they will be joining us on the stage, and then we will move on. And Jason Wakeland-Smith will be the moderator for the next session. Thank you very much.
So yes, I'm back again, but luckily this time around I don't have to give a presentation. So it's my great pleasure to introduce a couple of colleagues. You'll notice there's only one on the stage. I haven't lost somebody. Emma's joining us via a pre-recorded presentation, but I'll come back to that in a little bit. But just to introduce my colleagues then. So Doug Pham is the Associate Director for Clinical Policy in the Office of Study Integrity and Surveillance in the FDA's Center for Drug Evaluation and Research. He received a pharmacy degree from Oregon State University and his law degree from the University of Oregon. Doug recently joined OSIS after 10 years in CEDA's Office of Compliance and is currently focused on developing and improving the clinical BABE inspection program. The topic today is about the clinical study conduct side of things, so that's the focus for this session. In terms of Emma, I'll introduce her now. So Emma Whale is a Senior GCP and GLP Inspector for the MHRA in the UK. In her role as a senior inspector, Emma conducts inspections of all sorts of things, both within the GCP team, but also within the laboratories team. So that includes both commercial and non-commercial organisations in the UK and overseas inspections for bioequivalence and biosimilars. And at that point, I'd like to introduce Doug to the stage.
All right. Thank you, Jason, for the introduction. Good morning, everybody. It's an absolute honor and privilege to be here with you. As mentioned, my name is Doug Pham. Currently serve as the Associate Director for Clinical Policy within OSIS. After a number of years on the enforcement side of things, my current role allows me to kind of take a step back and really look at how we can improve the clinical trial conduct within the BE/BA space. More specifically, we want to be able to see if we can do it more efficiently, do it better, while also adhering to the kind of the pillars of what we want to do, especially in ensuring quality data, and then as well as protecting the human subjects that participate in these trials. With my brief time today, I just want to cover some of the basics about GCP principles, especially focused on documentation as well as informed consents, why they're important, and some of the concerning things that we've been seeing most recently. I know that in the past 2 days, we've been talking about the future of clinical trials and what kind of things we can do to innovate. But I'm going to be that guy on the last day of the conference to kind of pump the brakes a little bit and say, we're going to get back to basics and see if we can do things better today. Okay, so keep in mind that today's conversation is not about who did what or, oh man, I hope that wasn't me. It's just more of a, let's start a conversation and see if we can improve all of this together. This one. Okay. Okay. Disclaimer slide. I know that some of my colleagues have kind of passed through this, but with my legal background, I'm going to pause longer, especially with my inevitable oops sometimes, right? So, okay. So for today, we're just going to talk about a little bit of overview of FDA's BIMO program, talk a bit about source documentation, subject eligibility, with some examples and trends that we've been seeing. And finally, hope to cover some information about basic informed consent content requirements, as well as some examples and trends that we've been seeing. Okay, I know that we've already gone through this slide, but just briefly, OSIS sits within CDER under the umbrella of Office of Translational Science. We're made up of 2 divisions. And we are heavily interconnected with all the other offices within CDER. And we align very closely with Office New Drugs, Office Generic Drugs, Office Compliance, and we also work really closely with our counterparts in the Office of Regulatory Affairs. Okay, from a very simplistic perspective, OSIS performs 4 basic functions. The first, we select sites to inspect. The second, we perform the inspections themselves. Third, we report on those inspections via an established inspection report and EIR reviews. And lastly, we support CDER by providing recommendations on data reliability as well as compliance evaluations. To note, when I talk about the clinical side of things within OSIS, we tend to veer more toward both site selection and then doing the support of CDER with our recommendations, partly because ORA does all of our inspections and RRAs. Okay, for FDA's BIMO program is a comprehensive program. I'm sure that you're all aware of it, right? It includes inspections, data audits, and it's designed to monitor all aspects of FDA-regulated research. The BIMO programs are essentially there to hit 2 primary things. First, to assure the quality of the data that's submitted in any application. And second, to protect the human rights, safety, and welfare of those subjects or participants that participate in the clinical trials. There are currently 11 BIMO programs. They span from IRBs to clinical investigators to sponsors, in addition to in vivo BA/BE studies. For today, we're just going to focus on the in vivo BA/BE, specifically compliance program 7348.003. Please keep in mind that these CPs or compliance programs are essentially instructions to FDA personnel and compliance activities, but for everybody else, it's a great source of information about what we're looking at, what we want to expect to see, and it's really helpful for your clinical sites to kind of take a look at and make sure you're kind of in line with these compliance programs. Okay, before we take a deep dive in the CP, let's talk about why documentation is so important. Much like all our previous speakers and our colleagues, documentation is kind of the cornerstone of how we reconstruct data or studies, how we assess study conduct, and how we determine data integrity as part of data acceptability in any pending application. Without kind of overstating things, documentation is absolutely vital, right? In compliance program 7348.003, there's a really good discussion on informed consent as well as study source records. And specific to the studies or subject records, a clinical site should have documentation that demonstrates that a subject met eligibility criteria. A clinical site should have documentation that accurately or demonstrates accurate and timely reporting of adverse events, as well as serious adverse events. And a clinical site should document or have documentation that enrolled subjects are alive, real, and actively participating. One thing about subject eligibility, it's really important to have source documents to show that the subject has met all inclusion criteria. Without records to show that each criteria was met, the assumptions that they haven't been met. For each type of records, here we're looking at record organization, condition, accessibility, and completeness, your typical ALCOA standards. Okay, the first example that's been kind of popping up here and there is this absolute lack of source documentation to show subject eligibility. The example here speaks to a situation where there's no records to indicate a subject had achieved a certain dose of a specific drug within the 2-week period prior to enrollment. You can insert a variety of different factors. Here are subject eligibility requirements or fact scenarios. But in the end, if you have no records to affirm eligibility, then it's really difficult to say the subject is eligible. The second example here speaks to when you have conflicts in documentation. And here the example speaks to where the inclusion criteria requires a grade 3 of a particular acne assessment, but the records indicate both a grade 1 and a grade 3. While the contrast of records doesn't fall in that doomsday scenario like our first example where no records equals no eligibility, here the contrast or discrepancy creates a lot of question, right? To kind of help assess that, we have to dig a little bit deeper, and we consider a lot of different factors. The first is whether the record discrepancy is meaningful or impactful, right? So let's assume for this particular one, the protocol requires that the grade 3 acne diagnoses has to occur for, or must be within 30 days of enrollment in the study, right? And let's say, for example, that the grade 1 record was within a week of the grade 3. So in that scenario, it's kind of meaningful, right? Because you're shifting, the records contradict that need to meet that 30-day requirement. On the flip side though, let's say the grade 1 document was 10 years ago. So at that point, that record doesn't really mean much. Another concern to consider is whether there's an interplay with the study design and the study drug itself, right? There's a greater risk of impact when the study drug is intended to treat acne, but it's less so when it's not. And lastly, something to consider is what did the clinical site do or not do as a result of the discrepancy? So some things to think about that will help to kind of determine the impact of that record discrepancy. Okay, the last example here relates to documentation is when you do have documentation, but the records themselves do not clearly link to a specific subject. It's been described in a variety of different ways, and it typically comes down to where you have source records that are either unreadable, or the source records themselves don't have any link or attribution to a specific subject. While there's lots of factors, different types of explanations, from our perspective, you're kind of left with a lot of questions, right? On the one hand, you do have records, but on the other hand, without that link or attribution to specific subjects, it means you kind of don't have records, or it's not meaningful in that way. Okay, let's switch gears a little bit, and let's talk about informed consents. Within the CP 7348.003, there is tons of information on informed consents, and it speaks to a couple of different things, but it includes kind of the timing of when informed consents must be signed, who can sign, and the contents within the ICF form itself, with special reference to ICHE 6 as well as 21 CFR Part 50. And when we talk about 21 CFR Part 50, 5025 discusses some of the basic tenets about what should be included within the informed consent forms itself, right? Amongst kind of a laundry list of things, it should include a statement that you're participating in a clinical trial, the purpose of the study itself, and the expectations while you're in the study. As you'll see with some of the examples, there's a bit of a concerning trend of late where the informed consents used in BA, BE studies are kind of wonky, they're weird, they're a little confusing, right? And these odd statements in the ICF itself creates a lot of confusion as to what the subject is enrolling into, as well as the purposes of the study itself. So let's take a look. Okay, the first example speaks to a trend that we've been seeing a lot in ICFs. The typical language used goes something like this. The study is a clinical research project, but no part of it is of experimental nature. So that's kind of my first reaction too. When you first read it, you're like, it's kind of weird, right? Read it a couple of times and they're like, you know what, that's downright contradictory. But despite kind of that, all that, there's curious rationales for this that we've been getting. That the statement in these ICFs, they range that this is a template we're using, or this is a BE study, so the drug's approved, so it makes sense in a way. But regardless, though, a statement like that in your ICF is kind of confusing. And if you're confused like I am, then we kind of start down that rabbit hole of like, is it even appropriate to be in the ICF? Much like the first, the second example creates a lack of clarity as to the risks associated with the enrollment in the study. The language we've been seeing includes something like this. The drug has been on the market for quite some years, and all aspects of its usage are well studied. There are no unforeseeable risks associated with it. Okay, well, I can understand the statements because, it's a BE study, it may be an approved product that you're looking at, but the statements in itself, in an ICF, creates a lot of confusion and may be interpreted as downplaying the risks. Okay, so seems like we've hit the end of the road. That was a really brief chat on the basics of, source documentation, subject eligibility, as well as informed consents and the contents within it. What I want to highlight, or at least remind folks, is that yes, we want to look toward the future and innovate and do better in our clinical trials, but we do want to make sure that we do the basic things correctly and well. To close, because my house is full boys, and we kind of skip the Disney movies, I'll end with a quote from Big Daddy in the Adam Sandler movie from Sonny Koufax. He says, "Having a kid is great as long as his eyes are closed and he's not moving or speaking." So in that vein, take care of your house. Do all the things you're supposed to do. Thank you.
So the next part of this is to introduce Emma's recorded presentation. So don't worry, Doug and I are going to leave the stage and you're going to see us walk towards the back. We will come back, don't worry, we'll be back after Emma's presentation along with some special guests. But more of that in a minute. Okay, thank you.
Thank you, and good morning, good afternoon, everyone. Thank you for the opportunity to present to you during this event, and apologies that I can't be there in person. I want to use this time to talk through some common issues, inspection findings and case studies identified during recent MHRA inspections of the clinical portion of bioequivalence studies. Michael has already shown this slide earlier today, but it summarises nicely what we've done in terms of bioequivalence inspections since 2019 and the different approaches that we're able to use for our inspections. I wanted to include this here to reiterate that these different inspection approaches apply to both the clinical and the analytical elements of our inspections. We haven't yet had an example of an inspection where the clinical part has been done remotely and the analytical part has been performed on site, or the other way around, but we would be able to adopt this approach if it was deemed to be necessary for a particular inspection or a particular study. This next slide has also been presented earlier today and shows you some additional detail relating to the critical and major findings identified during our inspections. Where Michael spoke to you earlier today about the study activities highlighted in blue on this slide, I'm going to be talking through those highlighted in orange. So the critical findings relating to the clinic, only one of the 4 on the slide relating to subject eligibility, and the major findings presented on the right-hand side of the slide in the orange box. The findings that aren't in a box haven't been forgotten, they relate more to general supporting systems. This next summary slide shows a very colourful breakdown of the other findings identified during our inspections. I don't want to go into too much detail here, but the diagram represents the other finding categories that have been identified on 5 or more occasions during our inspections. And I just wanted to acknowledge that there's a pretty even distribution between the different areas. It isn't that we have one particular problem area. And the list on the right-hand side of the slide shows the other finding categories that have been identified but less frequently. Okay, moving on now, I want to use the rest of my time in this presentation to talk through some of the areas in the clinic where we're still seeing recurring issues and where significant findings are still quite common, just to raise awareness of the types of things that we're encountering. Hopefully, if these are things that you're familiar with in your organisation, you may be able to put measures in place to avoid some of these findings in the future, or to avoid some of the business and compliance challenges that these situations can present. So these are the main areas that I'll be covering. Let's move on to the first one. Often during our inspections, we encounter clinical pathology labs that are external to the company that we're inspecting. In these situations, we look at the vendor assessment process and the written agreements that are in place that describe the services to be provided, the division of responsibilities and so on. And we often find issues here. Either the contracted lab failed to deliver the services in the contract, or the contract disagrees with the clinical protocol or design for particular studies in the nature of the sample analysis to be performed. We've seen situations where a bioequivalence facility has contracts with multiple labs as a backup plan, which makes sense from a business continuity point of view. But where we've seen issues with this is where multiple labs are used within the same study, and their approach to sample analysis and the associated reference ranges for lab testing are different. So a result from one subject tested at one laboratory may be acceptable, but if the same sample had been sent to a second laboratory with a different set of reference ranges, the result may have been out of range. In these situations, it makes it very difficult to interpret and to compare the results from subject samples side by side, where they've been assessed using different reference ranges in different laboratories. We still see companies that use an external contract pathology lab with a set of reference ranges, but the Bioequivalence Centre also have a set of reference ranges as an appendix to one of their in-house SOPs. Often these sets of ranges are not the same, which presents issues when there are results that are acceptable according to one, but not acceptable according to another, and it's not clear how these results should be handled. We've also seen stability times applied to samples prior to analysis that haven't been met. So urine samples sent to an external lab and tested outside the established stability period, with the results being used to determine subject eligibility. The issue here isn't just with the potential impact on subject recruitment, but also the relationship between the external lab and the Bioequivalence Centre, as the lab would have been aware of the stability limitations of their own assay and the sample collection times from the clinic, but there's no communication with the clinic or a mechanism put in place to ensure that samples are received with sufficient time to ensure accurate analysis. Also relating to clinical pathology labs, we're still seeing issues with assessment of clinical significance of results. Either these assessments haven't been done for out-of-range results, or they're done for laboratory tests where the protocol doesn't permit any assessment or interpretation. So there's a hard limit on what the results need to be, either between 2 values or less than a certain limit. But the results are assessed as not clinically significant and the subject is recruited regardless. We also see laboratory testing for parameters that aren't detailed in the trial protocol. Often this is driven by the pathology lab, where they run a panel of tests regardless of what the sample is for. And in this situation, we would look at communication between the bioequivalence centre and the lab to ensure that protocol compliance issues are actively avoided. This can also present a subject welfare issue, as you're potentially testing subject samples for parameters that they've not agreed to via the subject consent process, and how you handle any results that come back indicating there may be something wrong when you didn't have consent to run those tests in the first place, particularly if it's something sensitive. I've also put eligibility issues on the slide, but I think I've covered those already. So moving on, the next area I wanted to talk about is IMP management. We've seen an increase in the number of findings relating to certificates of analysis for products being tested in bioequivalence studies. We still see where there's no expiry date available for the test product, either at the time of dosing, and I think once we saw where this was listed as not applicable in the final report for a study, which presents a significant subject safety concern and potentially ethical issues where subsequent stability is not established. I have seen on a number of occasions where the results listed on the certificate of analysis for product testing versus predefined specifications have indicated out of range results, or where testing hasn't been done, but these gaps and discrepancies haven't been questioned or challenged prior to study dosing. We've also seen where provisional data has been included in certificates of analysis, essentially draft data that is subject to change, but this has also not been challenged or addressed prior to dosing. I've seen issues with product verification checks, where these have identified discrepancies between what the product is versus what it should be, but these discrepancies haven't been adequately resolved prior to dosing. I've had many discussions on inspections about the difference between white, off-white, pale yellow tablets and the potential impact of these discrepancies and how to handle them. I think the advice would be, if you are in any doubt that what you've been provided with is not what you're expecting to receive, then do not go ahead with dosing. And then my final 2 points on IMP management relate to data loggers, where these are included in shipments of products to be tested in bioequivalence studies. You need to ensure that you have access to the data that they hold to assure yourselves that the products have been shipped appropriately and that they're suitable for use in the study. Often the software needed to retrieve the data isn't available at the bioequivalence centre, and the data loggers are sent back to the delivery centre, never to be seen again. And this can present a significant problem where the product has been transported globally over a long duration with specific storage requirements. Also, where you're provided with the downloaded data, you need to be able to link this directly to the shipment that it related to. Being able to approximate that the date range looks about right isn't really adequate for a temperature-sensitive, unlicensed medicine that's about to be administered to humans. And finally, we've seen issues recently for facilities that may have well-established systems and processes for studies involving oral tablet dosing, but when they move into new study types for different IMP formulations, they encounter issues specifically relating to IMP, such as validity of supporting paperwork, verification checks, and accountability, and so on. In these situations, we'd advise that you prospectively look at your existing processes and pull them apart to understand what purpose they serve and how this can be replicated for the new study type, and introduce new steps, as it's highly unlikely your existing ways of working can be directly applied to a new study type without any modifications. Moving on now to talk about issues we've seen relating to computerized systems. These are being introduced more and more in bioequivalence studies, and we welcome this approach where it streamlines things and makes things easier, but they do come with their own set of challenges. The main areas we encounter findings are electronic case report forms and volunteer recruitment databases, so we'll look at these two areas in turn. Starting with ECRFs, we see all different kinds of systems being used, from those developed entirely in-house to those bought off the shelf from a vendor, and the documentation supporting their development, testing and release for each bioequivalence study varies hugely. Some of the more common issues we see relate to audit trail functionality in these systems. Often the data capture side of things is pretty good. It will let you enter the data you need, otherwise you wouldn't have chose to use it. But the ability to see who did what in the system, when they did it, and the ability to track any changes in the data is often lacking. Many systems have little or no audit trail functionality, and comments in the audit trail such as data update or record modified are not particularly useful and wouldn't help your case if that was all that was available to defend a data change, particularly if the changes are also not date and time stamped and can't be attributed to an individual. Essentially, when you're looking for an ECRF solution, we'd encourage you to look into the audit trail functionality in some detail. We also see systems not permitting study-specific builds or historic systems that aren't fit for purpose, where subject data is forced to fit into existing data fields that they're not designed for. These can take a lot of time to understand or interpret, and the data can easily be misrepresented. Barcoding is becoming more and more common. We've seen, we have some clinics in the UK where there's very little manual data entry in the ECRF system, and everything used in the trial has a barcode, so staff, participants, equipment, and everything gets scanned as study activities are occurring. These systems are fine, and this approach reduces the risk of manual data entry errors, but they can be abused, where the relevant barcodes are copied and stuck on the front of a study folder and scanned from there, rather than the system being used as intended. And there are also examples of ECRF study building and testing in a live environment and not a sandbox or a test environment, which can present a risk to live system data integrity where things can be edited in error. Moving on to volunteer databases, the main issues here relate to data migration from old to new systems. We've seen a lot of these recently, and often the migration's incomplete, where not all data has been migrated as intended and the gaps haven't been identified. Also, the detail in documentation relating to how the migration was managed is often lacking and doesn't provide much confidence that the migration effort was comprehensive and successful. And then finally, biometrics. We're seeing these more and more in bioequivalence centres for identifying volunteers at clinic visits using fingerprint scanners. We've had a few issues with these over the years. Often the scanners fail to identify the correct individual, or the same fingerprint scanned multiple times identifies a number of different individuals, suggesting an issue with the specificity of the scanners. This obviously raises significant concerns when this approach is used as the primary mechanism for identifying subjects on arrival at the clinic. More and more often, we're seeing clinics that have the capability for biometrics, but they're not using them, which suggests that they don't trust them. Okay, the next area to talk about is facilities and equipment. Most of the findings we've identified in this area relate to facility washrooms and alarm points. During my time as an inspector, I've been locked in many washrooms in bioequivalence centres and challenged staff to retrieve me from behind locked doors. While this is quite a light-hearted story, the point of the exercise is to demonstrate that washroom design doesn't always allow for access to trial participants behind a locked door in the event of a collapse. In response to this, we've also performed inspections to the soundtrack of electric saws, drilling and hammering as facility upgrades are being made, demonstrating that often these facility issues are easy to fix and commonly are resolved before our inspections are completed. Regarding alarm points, we look at the location of these throughout the BE facility during our inspections, and we look at the testing schedule for them to ensure that this is done regularly and it's done successfully. With bioequivalence trials, there tends to be a feeling that the products being tested are generics with well understood safety profiles, and mechanisms for management of medical emergencies of lesser importance, but facilities should always be prepared for these to occur, and this is often not the case. Also on this slide, I've mentioned equipment maintenance and calibration. just to highlight a few areas where we've seen issues with the frequency and effectiveness of equipment maintenance. Testing kits such as urine, drugs of abuse and pregnancy testing kits being stored at incorrect temperatures, and communication issues with automated interfaces between equipment and the ECRF where these are in use. Okay, moving on to talk about electronic archiving. We've raised many findings over the years relating to archiving electronic data, and often it isn't the data itself that's the issue, but the metadata that sits behind it. I've listed on the slide some of the systems where consideration should be given to ensuring any related metadata is also archived where there's electronic data available. By metadata, I mean any settings, parameters, supporting information that's held within the system that you need access to in order to make sense of the data itself. So settings on an ECG machine or an X-ray machine, or any supporting information that's not presented as part of the data itself, and where the data would be incomplete without it. And then the final area I want to touch on before I move on is to talk about insurance. During our inspections, we're asked to review a copy of your clinical trials insurance policy. And over the years, we've seen a number of issues with the type of cover in place, ranging from specific populations being excluded from policy cover, specific global regions being excluded, specific medicines being excluded, which could be an issue for future trials where you may not know what products you'd be asked to work on, and specific types of studies being excluded. Also, we've seen gaps between the end of one policy and the start of the next policy where trial work has been ongoing. I haven't yet seen a situation where there's been a need for an insurance claim, where there's been a gap in cover, but I think that's been more fortuitous than anything. So I would urge you to check the terms of your clinical trials insurance, the duration of your policies, and also any exclusions in your policies that you may not be aware of when you next have the opportunity. Okay. The final area I wanted to quickly move on to is to talk about transparency in bioequivalence studies. This issue could apply to any study documentation and any activity in a study, and it really relates to how representative are the study records of what has actually occurred. We see many situations where the way things are recorded do not accurately represent what took place, and the time and resource needed to get to the bottom of things and fully understand what took place is wasteful and unnecessary. It tends to be around unusual events, investigations, interpretation of data and deviations, and I have a few case studies to go through which will help illustrate my point. But when you're involved in study activities, I want you to ask yourself, I know what I mean, but is that what it says? Do any assumptions need to be made regarding what's been written, and could this be misinterpreted? So this first example was from a recent inspection where we were told that QA were not listing all audits for certain types of studies on QA statements. The justification for this was that there were multiple audits that were relevant and the QA statements would be far too lengthy to list them all, so only a sample of audits were routinely included in the report. The issue here is that there was no qualifying comment on the QA statements to explain to the reader that there was additional audit information available. So it would appear to the reader that the listed audits were all that there was to demonstrate QA coverage for a study. Worst case scenario here is that an objective reviewer of the report, so an assessor at a receiving authority, for example, could determine that the QA coverage wasn't sufficient for the study, could ask more questions, which could lead to delays, and in worst case scenario, the data may be rejected. But if the data had been presented in an unambiguous, comprehensive manner, the situation could potentially be avoided. The second case study, paper records for a study activity indicated for several subjects ND, indicating that it hadn't been done, with some additional coding for the same procedure and the same subjects indicating subject non-compliance. On initial review, this suggested that the subjects had been non-compliant during the study, when in effect the procedure in question hadn't been performed by study staff and it was nothing to do with the subjects at all. So essentially the way the coding had been used in the data was misleading and not very transparent. This next example, exclusion of a study subject for other reasons. We see this quite often, and normally we just look into subject withdrawal, potentially for safety reasons that requires maybe further investigation if we think it's safety related. So other reasons could suggest it's a safety issue. On this occasion, only 27 of the planned 28 subjects had been recruited, as the study had sufficient statistical power without the data from the final individual. But justifying this approach in the data as other didn't really explain this in sufficient detail and prompted further questions and investigation that was time consuming and could have been avoided. Then this final example, 2 subjects were shown as not dosed in IMP data, but there was dosing data and subsequent clinical data available for both of them. It turned out that they had been dosed, just not on the date that dosing was originally intended to take place. Again, the time taken to investigate the issue, to arrange meetings with relevant staff to explain the issue was wasteful, and it could have been avoided if the method of documenting the situation had been clearer. So the take-home message from this part of my presentation is really to be clear that the way your studies are documented, your investigations are performed, are truly representative of what took place, to avoid any potential for misinterpretation, confusion, misunderstanding and unnecessary delays. Okay, thank you all for your time and for listening. I will leave it there and hand back to the room. If there are any questions relating to my slides, there are people in the room who will be able to take those during the panel session. So thank you again.
To bring up my special guests, so if I could bring up Jennifer Adams, Doug and Michael McGuinness, that would be great. So we're going to play this one a little bit by ear. We have some questions which have been submitted, but equally a little bit conscious that you've only just finished listening to the last one. So we may end up with a few more as they come through, and we'll see where we go. So the first question I have is to start with Michael. So Michael, how do you decide what studies to perform inspections on?
That's trade secrets. I mean, it's fair to say it's a logical process, but it depends on the study type. So we have in the UK routine and triggered inspections. For routine inspections, we will look at the list of studies that have been submitted to us, evaluate where they are of interest. If you've previously been inspected to us, there might be specific aspects of those study conduct which we find would best show your compliance and a suitable candidate. Triggered is very different, obviously, because it's been raised as part of an application process and it's very specific and targeted. So yeah, it's not one factor. So it's not like a case of the 4th one down on the list or anything like that.
So yes, it's a double secret, trade secret. But I mean, like MHRA, it's a multitude of factors that you consider, including, the study, past history, or, if we have additional information, or there's certain things about the application that we want to take a look at or not. So there's a variety of different things that comes into play as part of that calculus. But yeah, it's double secret.
So then following on from that a little bit, so Doug, can you talk a little bit about how, or does the FDA complete BIMO inspections for all NDAs and ANDAs?
Yeah, so we do not complete all inspections of NDAs and ANDAs. As part of the risk-based assessment, we try to do the best that we can, and we use it to site select, as mentioned before, using all the factors to go into whether it's an NDA or an ANDA. We're using a variety of factors to determine whether we go or not. Do we inspect in India? Yes, we do. A lot of our inspections are both NDAs and ANDAs. And there are a variety of different actors that we take a look at as well there too. So.
Thank you. Jenna, did you have anything to add on that one?
Sure, yeah. So The follow-up question to that was, do we conduct inspections from sponsors located in India? So we would inspect facilities regardless of the location of the sponsor if it was deemed necessary. That said, the bulk of our bioequivalence work is located in India. It's about a third of Office of Bioresearch Monitoring's total foreign work plan is spent within India. Specifically doing clinical bioequivalence inspections. I think it's fair to say that the analytical inspections, a lot of them are located in India probably as well.
Thank you. You can leave your mic on because I'm going to give the next one to you as well. So what's the role of paper source worksheets when the eligibility and the medical records and research activities are available in the subject's electronic medical records? For the person who asked the question, for their studies, they have the subject's medical record, ECRFs, and paper source worksheet. So they're worried a little bit about duplication of data.
And I think I would agree that this seems like a lot of duplicate entry. And anytime you have a single data point that's being recorded over and over again, you increase the risk of error and having discrepant data, which then requires more effort to explain. I would just say that, you know, if you wanted to eliminate the middleman, in this instance, the paper source worksheets, you just would want to make sure you take a careful look at your EMR and make sure that it has all of the information that you actually want to capture, that it's accurate, that it's complete, and that it has adequate controls on the system. The last thing you would want is for somebody totally uninvolved in your study to be able to delete records that you are relying on for your source. So I would just make sure that you do your due diligence when assessing your EMR, but then I don't see any added value in transcribing that data yet. Again, you're only going to introduce more error.
Thank you. Michael, did you have anything to add on that one?
Nothing, just to echo the, obviously when you record something more than once, then it's definitely a risk factor. But yeah, I think for the sake of time.
Excellent, thank you. So next question, I'm going to hand it to Doug first of all. Do the inspectors look at the data generated, so things like pre and post study safety assessments by the clinical path labs when you're looking at BE studies?
The short answer is yes, we do. And you know, we'll take a look at a lot of different things, and this is part of that kind of area that we can focus on and take a look at. But you know, it's not going to be the end all be all. We take a lot. We look at that, but then we also weigh it against other things that we're looking at within the source documents or the study records as well. So.
And then Michael, do you want to chip into that one?
Yeah, I think Emma touched on this as part. The clinical laboratory files you usually get back can form part of subject eligibility, and you've seen the example that Emma discussed. So yes, we do review that. Emma mentioned that you do have both inbuilt pathology labs within the bioequivalence centres, but also external. So the level to which we review the data might be dependent on access, but certainly they form a key part of when we're doing that eligibility assessment. It's fundamental to subject safety to make sure you don't have anybody in the trial that's at possible risk.
Thank you. And I'll stick on the theme of clinical labs. This one is certainly something we've seen on inspection. So the question is, can clinical facilities have separate reference values for subject eligibility, which are different than the clinical laboratory reference, whether that's in an investigator's SOP or the facility's own list of values or protocol appendices? So perhaps you can kick that one off.
This one arrived late, so this is more off the cuff. The short answer is it will definitely depend. You can definitely see a scenario where a number of vendors are associated with a bioequivalence site, and the onboarding process will look at the difference between the laboratory reference ranges that those labs have and whether or not they make use of their in-house facilities, the in-house SOP-defined reference ranges. So one of the things that's crucial to consider on this point is inter-study, you're looking to compare apples with apples. So if you change a vendor during it, that is different to if you're using a single vendor for study A than when you might be using a different vendor for study B. We have, with bioequivalence centres, seen a variety of approaches to clinical reference ranges where the investigator's SOP overrides the accredited laboratory, usually expanding the acceptable ranges. And again, this is something that we'll query on inspection to look at what is that? Is that too ultimate goal is that to extend recruitment and make it easier, but equally, is there any risk to subject safety? So it very much depends on the setup. And if the question was submitted in the room, I'm more than happy to talk in greater detail about the specific aspect, but it's a very good question.
I think the other key bit to that is that you almost have to stick with one. You can't pick and choose depending on what you feel like. So if you use one person's values to get somebody in and then use another set of values to keep somebody in potentially later on. So pick and stick with one. I couldn't help myself but give myself a question. I do apologise. Sorry, gang. So the question is, for the protection of human participants in a clinical trial or a BE study, is an insurance policy compulsory to come from the sponsor? So is it fine if there's no insurance policy, but the sponsor includes a statement in the master service agreement saying they'll cover any related injuries due to the study? In essence, that's a flat no. There's no self-insurance permitted. Doesn't matter who you are or what you have. That doesn't fly. You'll always need an insurance policy to cover your clinical trial. And you also need to make sure that there are no exclusions for each protocol that you run underneath it. Certainly, we've matched them up over the years and found trials which the insurance policy says you either can't be doing trials of this medication or in a particular group of participants. And lo and behold, that's exactly the kind of trials that we've found. The other thing is just to look for dates and that they actually cover the activities and the sites and things that you have listed. So insurance is always an interesting one, not particularly exciting to look through, but there's often plenty of things to find within it. The next question is for Doug. What's expected in terms of proof within source documents of not meeting the exclusion criteria? And in particular studies which don't have chronic diseases, past medical records, those kind of things.
Yeah, so most of the conversation that I had before was talking about inclusion, so where you have to have something before you can come in. When we're talking about exclusion criteria, it's a little bit different perspective. It's what things would take you out of the pool of becoming part of the trial. We would look at the documentation as you screen, right? As what kind of questions are you looking at, or what kind of things are you talking to the subjects around, get a medical history, especially if it's like, let's say an easy one would be an exclusion because you're a diabetic, right? You would expect the CI or the clinical site to pull a history at least to kind of take a look and then start going through the interview and doing the checkoffs to make sure that they are not a diabetic prior to enrollment. So the exclusion side is not as clear cut as having to show something, but it's also part process and part of your self-created documentation.
Thank you. Michael, did you want to add anything to that one?
No, it's just to say that it's very difficult to prove a negative. Within the clinical documentation, we do see, for example, text to say that check of exclusion hasn't been met. Particularly for bioequivalence centres, we do see it, but again, the level of documentation that goes in the background, it's something that we would check.
Thank you. The next one's to all of you, and it's sort of a link across all types of trials, really. So would you all agree that most observations in BE inspections are similar to investigational clinical trials? I'll give it to you, Jenna, seeing as you've got that puzzled look on your face.
I mean, the objective of the program is the same: human subject protection and data integrity. So I would say that, because those are the focuses, regardless of whether we're doing a new molecular entity study versus a bioequivalence study, we're going to find things in those same two buckets regardless. I don't know if I'll throw it to Doug.
So I would agree. I think those are the primary things, to ensuring the quality of the data that's generated at the site, versus, or on the same vein, is protecting those subjects that are enrolled in that site. How you get about that will primarily depend on the functions of the site, the design of the study, and the protocol itself. And those details are going to be kind of driven by that aspect of it. But the overall approach, depending, regardless of a BE study or any other type of study, it's going to be very similar.
Michael?
Yep, so fully agree with my FDA colleagues. It's interesting because we have the same objective. As we said, the inspector's aim is there to find anything that detracts from data validity or anything where the welfare of the subject has been put at risk. It's fair to say, though, to expand that across the GXPs. I have the advantage of being in a multi-GXP team, and we share everything between us, the observations, people's, like we say, whether it's conducted in Glasgow or here in Maryland, people have similar traits, and it's taking those learnings and applying it across the board. I wouldn't restrict it to say the presentation topics we've heard today don't apply to you because you don't do generic applications, but you never stop learning.
I'll chip in a little bit as well. I think BE studies are a little bit different in some respects because they're so compact and they're so intrinsically linked. You have your participants, you dose them, you take some samples, you sit on those samples for a little bit while you finish the rest of them, and then you analyse them all. The trials in the wider world with the patient populations take years to get to that point, and they're huge generally, whereas bioequivalence trials can be 40, 50, 60, 70 participants. So they're very different in terms of a speed kind of thing and that intrinsic link, and both parts are often done by the same organisation. So they do pose questions sometimes around access to data, the ability to look at things in different ways. Unlike some of the monster normal, in inverted commas, clinical trials that we see where there's thousands of vendors involved, multiple sites, multiple investigators, all these kind of things. So there are different things to think about as well. On top of, is the data good? Are the participants safe? Can we trust the data? So there are nuances to all of these things. Again, I'll throw the next one open to the group. Can you tell me a little bit about sort of collaboration for sharing observations across GXPs? And I'll widen the question and say about, is this being about collaboration between us as organisations? So can you talk to me, first of all, about collaboration within your organisation, and then secondly, about collaboration between us as a group?
I can kick us off and then happy for. So yeah, in terms of, if I've got this right, collaboration within our group. So as I said, we share everything that we talk about. We have team meetings. We try to ensure consistency between inspectors, so it doesn't matter whether you get myself, Jason, Emma turning up, that you get a similar approach. So collaboration through training, discussions, professional development is absolutely there. All of our reports are peer reviewed by a colleague that was independent of the inspection itself. So in terms of that aspect, it's something that we encourage. It's well supported by management. In terms of collaboration across the different agencies, if I've not skipped Jason's question, I think it's very true that you've heard almost for the last 3 days, the word collaboration has been the word of the workshop. We work very closely with our partners here in North America, both FDA and Health Canada, and it's fair to say that information is invaluable. Lessons learned, lessons shared. Again, we talked about the similarities of GCP inspections, the specific aspects of BE, and we hold quarterly meetings between ourselves. I'm very familiar with colleagues here at FDA, but also at Health Canada, and that information sharing is an excellent source. And then I think if I open it up at that point.
Yep.
So on the FDA side, we are not the same group. We have the disadvantage of being a separate program. FDA aligned our programs, I think, in 2019. So our pharma folks are actually in a separate program from our bioma folks. That said, when you conduct an inspection, part of your preparation for the inspection is looking at the inspectional history, and the database of all of our inspectional findings is shared across all programs. And so we have access on a BIMO inspection if I'm inspecting a large bioequivalence facility that is also inspected under GMP. I have access to the full report, all of the observations, the names of the investigators who conduct it, and would have access to them and all of the data that they discovered on their GMP inspection to help inform my upcoming BIMO inspection. not much to add, but it's just in the case of FDA, the volume is kind of
an ordeal, right? And so we continue to collaborate with all the various offices, with ORA, within CEDAR, but it's just a huge amount of information and lots of inspections, lots of different types of inspections. But they do get pooled, and it is part of the calculus every time we site select, or when we go out to inspect, or when we do a review post-inspection. We're always looking at the totality of information. So that's a continuing thing, to continue the collaborations cross agencies. That's something we do all the time, and it's super fun. I think it's great, and it's a great way to share stories, share information, and like you mentioned, kind of learning moments about what you've seen here, certain types of inspections or different kind of things. So it's a valuable thing that I think has been going on for a bit of time, and it's something that I think everybody here is looking forward to continuing for the long term.
Thank you very much. I'm going to keep the next one because the answer is a bit of a negative, but the question was, is informed consent required for data taken from a biorepository? And we've taken that as sort of registry data where access to the patient may not be possible. So you don't know when the patient may next be coming in for a checkup, the patient's moved and didn't tell us, these kind of questions. So we've taken that as a BE kind of question, that you've had a volunteer for a while and perhaps they haven't attended the last visit. And we've taken it as, could you then use registry data to potentially follow them up if they just didn't come back? The answer in some respects is we have no experience of that, of using registry data in bioequivalence trials, really. We'd want that clearly defined if ever you were going to do that. And it seems to be a fairly blunt instrument of, are they still alive? Have they attended a hospital? And quite when you might be able to access that data, and even whether or not you have consent to do that. But just, I guess, be careful, but something to explore potentially in the future. I'm just going to give my poor panel one more question, and then I'm going to release you. The question's suitably broad, so by all means, feel free to go with it. The question is, can you please define oversight, study, and reporting? And so, Doug, I'll start with you.
Okay. So, oversight of study and reporting is, as you can imagine, huge, because it's, you're looking at different actors as part of the study. You're talking about the sponsor, you're talking about clinical sites, you're talking about clinical investigators, SSPs, CROs that are all in between. But what I would reiterate is that at every point or any touches within the study, you should have some overall oversight with what you're doing. And if you're the sponsor, you should have oversight in general of the entire study that's being performed. Those kind of controls should be built in a sense. It'll help prevent a lot of problems. It'll help, if there are problems, you've already pre-planned for them in how you approach them and deal with them. But the answer to this is, it kind of depends, right? Depends who you are and what you have to do. And then the reporting aspect of it will be related to that too. So it's a long-winded answer for me. Michael?
Yeah, I can go next. So as Doug said, it's a huge, huge piece. It's in terms of a bioequivalence study, the oversight, one of the key pieces is, like we talked about, those touch points. It's not, we've got screening, we've got then getting them in, getting subjects or trial participants into the clinic. Linking them up with something like a lab where you might have a light-sensitive molecule. Not having that two-way conversation, it's the same as pretty much any aspect of business. Communication is key. Oversight and control is a different aspect. Demonstrating it works differently in almost every company I've worked in or I've inspected. Some go with what I would call very militant, restrictive. We will meet every day at 9 o'clock. We'll have a toolbox meeting. We will go through this. Some don't. And there are strengths and weaknesses to both parts. The communication element is key, having information. The one thing is just to acknowledge the negative part of that. If you're a sponsor and you have outsourced everything to a BE centre, be aware of the role in which you as the sponsor are exerting your pressure and control. You looked during my bioanalytical presentation, if you'll allow me to jump into it, we saw the negative aspect of pressure on individuals. where they pushed, and then we ended up with a data integrity breach, which then resulted in the trial ultimately going down. So what might start off as positive oversight, positive control, can very quickly be turned into a negative, and that's a very slippery slope to try and climb back up from.
Jenna, did you want to add anything?
I mean, I guess I will highlight that this is a great question. We don't spend a lot of time talking about oversight in bioequivalence compared to the first 2 days of this course. But ultimately, the oversight of the study at all of the levels that Doug has discussed, the oversight by the clinical investigator, oversight by the sponsor, oversight by the CRO, all of these different levels of oversight have a direct influence on the integrity of the data and the protection of the human subjects that come out of the study. And so as a result, it's just as important in bioequivalence as it is in a normal non-BE clinical trial.
And I'd like to link it back to Emma's presentation in some respects, picking up the reporting side of things. So it's important that the report bears resemblance to what actually happened on the trial. We've seen plenty of examples in the past where there are an errant cut and paste job with particular issues, so this kind of thing, or you find things within the trial data itself which just don't appear in the clinical study report. And even when you're talking to people about it, they just don't necessarily appear. What's really important, and we talked about risk earlier on in the workshop, if things happen, if you tell us, we can think about it and give a considered opinion and try and link it up to how it all fits together and how it works. If you don't tell us and we find it, we've not really got too many options left to us. It might be possible to extend the inspection, but certainly if we're overseas, that's limited. Or the next one is to go back to the assessors and start to say, we have problems here. We found this stuff, it's not in the clinical study report. Over to you. What do you want to do with it? So be open, be honest, discuss, comment on what you found, talk about what you did when you found it and what you then put in place to try and deal with it. And then we can have an active conversation about what that is. That's really helpful to have it up front so we can prepare for it, talk to the assessors potentially if it's in the CSR. If you've omitted to put it in an error and it comes up in inspection, then again, talk to us early. Let us know that there's something we need to work our way through. So I'm going to draw it to a close there. Thank you very much, panel. Thank you very much for coming to join me on stage. If I could ask you to put your hands together and thank the attendees. My final job is the important one, and that's to let you all go for lunch. So we're back at 1 o'clock for lunch, so that gives you a bonus 12 minutes to grab a little bit of lunch. So thank you very much. We'll see you for pharmacovigilance this afternoon.